Imagine a fuel tank has leaked at an industrial site. The soil beneath is stained and smells of diesel. But how do we prove it and identify exactly what's in there? It starts with collecting a soil sample. But before we can analyze it, we need to extract the contaminants from the soil matrix. The soil is mixed with a solvent, usually dichloromethane or hexane, which pulls the hydrocarbon contaminants out of the soil and into a liquid extract. This extract is then ready for instrumental analysis. Our first technique is gas chromatography with a flame ionization detector, or GC-FID. The extract is injected into the GC column. Inside, compounds separate based on their boiling points and how they interact with the column's stationary phase. Lighter compounds travel faster. Heavier ones take longer. As each compound exits the column, it is burned in a hydrogen-air flame inside the detector. This generates an electrical signal, and every compound produces a peak on the chromatogram. What we see is a characteristic diesel fingerprint, a broad hump representing hundreds of unresolved hydrocarbons with distinct sharp peaks for individual N-alkanes from C10 all the way to C25. This pattern is instantly recognizable as diesel fuel contamination. GC-FID confirms contamination is present and tells us how much. It is fast, sensitive, and cost-effective, perfect for initial screening. But to know exactly what compounds are present, we need to go one step further. Gas chromatography mass spectrometry, or GC-MS. In GC-MS, compounds leaving the GC column are bombarded with electrons. This breaks each molecule into a unique pattern of fragments, a molecular fingerprint called a mass spectrum. This spectrum is compared against reference libraries containing hundreds of thousands of known compounds. The result is a precise, confirmed identification of each chemical in the sample. GC-MS can identify specific toxic compounds like polycyclic aromatic hydrocarbons, naphthalene, fluorine, and pyrene, which are regulated under environmental law due to their health risks. It can even determine the source of the contamination. By examining specific hydrocarbon ratios like pristane to phytane, forensic environmental scientists can distinguish diesel from petrol, kerosene, or other petroleum products. So to summarize, GC-FID is our first line of defense. It is fast and cost-effective, confirming diesel contamination and measuring total hydrocarbon levels. GC-MS is our precision tool, identifying individual compounds, meeting regulatory requirements, and supporting forensic investigation. Together, these two powerful techniques give environmental scientists everything they need, from initial detection to precise identification, helping to protect our soils, our water, and our communities.